Okay, welcome back. In this lecture, we're going to start our discussion of antibody applications. So what are some of the things that we can use antibodies for? What kind of technologies are served by our understanding of antibody structure and function? Well, a first question is, if we want to use antibodies, how do we make them? How do we get antibodies? Clearly we've seen how antibodies are produced during an immune response, but if we want to use them for some sort of applied purpose, what do we do? Well, it's pretty simple. We take an animal and we immunize it with an antigen of interest. So if we want to make an antibody, we need to know what we want the antibody to bind to, its antigen. We take some purified, tube or solution of that antibody and use it to immunize an animal. Often this is a rabbit. Rabbits are very common animals to make antibodies in, but it might be a mouse, a rat, a goat. There are other options. You immunize the rabbit, you just inject it with the antigen. It makes B cells and plasma cells and ultimately starts secreting antibodies into the serum. So you let the antibody production happen in the rabbit, and then a few weeks later, once the process has really become established, you take the blood from that rabbit and you isolate the serum. So you take out the cells, and the serum is going to contain a lot of antibodies against whatever you immunize it against. And so that serum, which contains the antibodies, is called antiserum for that reason. So this antiserum, if you'll notice here, I sort of have different colors of antibodies here. So this is recognizing that an antiserum is going to contain antibodies that recognize our antigen, but it's going to be a bunch of different types of antibodies. And by that, I don't mean antibody class. What I mean is that it's going to be antibodies that recognize the antigen on multiple locations on that antigen. So if you remember way back to our initial discussion of antibodies, all antibodies or all immunoglobulin molecules recognize antigens at a specific part, a specific motif or epitope on its tertiary structure, its shape. And as it turns out, antigens might have multiple locations where antibodies can actually bind. So in this example, we have a single antigen, but there are actually two antibodies here. One that recognizes this blue epitope, so the blue antibody, whereas the red antibody is recognizing the red epitope. So both of these antibodies have the same antigenic target, but they bind at a different physical location on the antigen itself. And so even though these share the same antigen, they are distinct antibodies made by different B cells. Remember, B cells only make a single antibody. So any individual B cell is going to make an antibody that only recognizes one single epitope. So we're going to need different B cell clones, different B cell populations, basically, in order to make antibodies against different epitopes. So we call antibodies, the collection of antibodies that recognize different epitopes on the same antigen, polyclonal antibodies for that reason. So poly meaning many, clonal, they come from multiple B cell clones, basically. So polyclonal antibodies recognize the same antigen on different epitopes. So you can see this here. If you have this sort of imaginary antigen and it has different epitopes here represented by a square, a triangle, and whatever this is, pentagon, you can see that our different colors of antibodies are recognizing these different epitopes on the surface of this antigen. However, sometimes, for clinical uses or for research uses, whatever, sometimes we want to make a pure stock of antibody that all recognizes one single epitope. We don't want it to bind all over the antigen. We want to bind one specific epitope for some reason. We can do that, and when we generate that population of antibodies which all recognize the same epitope, we call those monoclonal, and that's because all of these antibodies are coming from an identical B cell or plasma cell clone. So a tube of antibodies which all recognize the same epitope on an antigen would be called a collection of monoclonal antibodies. So how do we make monoclonal antibodies? If we just take the serum from our animal that we've immunized, it's going to contain polyclonal antibodies because, all sorts of B cells were being made. If we want to make monoclonal antibodies, we could take that serum and just use crude protein biochemistry to purify it out somehow. But that's not a very effective strategy. You would not get huge quantities using that approach. Also, you would have impurities. And in particular in therapeutic settings, sometimes we need a lot of monoclonal antibodies. So what are some strategies that we can use to generate monoclonal antibodies kind of on an industrial scale? Well, as it turns out, there's a very effective way to do this, and it involves creating what are called hybridomas. So, hybridoma is basically when we take some kind of animal, in this case, a very cursed looking mouse, we inject it with some antigen of interest that we want to make antibodies to. It has it has an immune response to the antigen and makes B cells and plasma cells, which start making antibody the way it normally would. What we do is we isolate the plasma cells from this animal and we fuse them or hybridize them together with a tumor cell. And so this tumor cell is an immortalized transformed cell, which will continue to divide forever. It's very metabolically active. In contrast to a plasma cell is terminally differentiated. It doesn't divide, and it certainly does not live forever. This is a way to take a plasma cell and make it highly proliferative and highly active. And when we do that, we form what's called a hybridoma. So oma typically indicates some kind of tumor cell. This hybridoma now is immortal. It'll keep dividing, and because it has the characteristics also of the plasma cell, it's going to make a whole bunch of antibodies. And because it came from a single plasma cell, it's going to make monoclonal antibodies because it all came from a single sort of mother plasma cell. And so all of the clones, the daughter cells of this plasma cell, which we use to make the hybridoma, are all going to make the same exact identical antibody. So they're monoclonal antibodies. If we look at this in just a little bit more detail, in this case, a slightly more normal looking mouse has been immunized with some kind of antigen. In this case, this antigen has 4 imaginary epitopes on it that antibodies could bind. So if we immunize this animal with this antigen, it's going to make 4 different types of antibodies, one that recognizes each of the epitopes. So you can see this antibody 1, 2, 3, and 4. And you can kind of see them binding to the various epitopes here. If we want to make monoclonal antibodies, though, what we're going to do is isolate plasma cells from this mouse. And so in this case, we would have 4 different types of plasma cells, each creating one of the types of antibodies that we have. We hybridize them with our myeloma cells or cancer cells. So we merge them, hybridize them into our hybridomas. Then what we do is we take that population of hybridomas and we dilute it really, really thin. So, we dilute it many fold until we get to such concentrations that we are confident that a single well in our culture dish, for example, contains one single hybridoma cell. So if it contains one single hybridoma cell, it can only be one clone, right? We do that and isolate individual, like literal one hybridoma cell at a time. And then once we do that, that cell will start to divide, and any daughter cells that are produced within that well all came from the original cell. So they're all clones of the original cell. And because they're a pure population of a single clone, the antibodies that they make are monoclonal. So this would be a way, if we wanted to make large concentrations of any of the original 4 types of antibodies that were produced by this mouse, we could go through this process in order to generate those monoclonal antibodies. In this process, again, we can do it in huge volumes. Once we isolate our hybridoma, we can then grow it up in huge vats, basically, and make enormous quantities of monoclonal antibodies using this technique. So why would we do that? One is that a lot of times things that we want to target with antibodies has, one antigen has some kind, or one epitope has some advantage over the others. Maybe it generates a stronger effector response if we use it therapeutically. Maybe something about that part of the epitope prevents it from binding to something else that we're trying to target. There are a lot of reasons, but some of the major clinical applications of monoclonal antibodies include things like neutralizing other ligands and receptors. And we've seen some examples of this already. So for example, there are a lot of different autoimmune disorders which have overactive inflammatory cytokine signaling components. One example there are a lot of patients who suffer from what we call interferonopathies. These are cases where patients are just making a lot of interferon when they shouldn't be. They don't have an infection, but through whatever genetic or environmental reason, they have interferon activation, which is causing autoimmune attack of their own tissues. And so a common way to prevent this is to treat them with monoclonal antibodies that have been raised against various components of the interferon signaling cascade. So if if you remember back to the last module, type 1 interferon has a couple of different ligands. Interferon alpha is one of them. It binds to the interferon alpha receptor, IFNAR, which induces all the signaling that we've discussed. So if we wanted to turn off interferon signaling to alleviate the symptoms of autoimmunity, we could use monoclonal antibodies that either recognize the ligand, interferon alpha, or perhaps the binding pocket of IFNAR, the receptor. In either case, antibodies are large molecules, so when you attach an antibody to something, usually that means that it can't bind to its normal partner after that. So, we could neutralize either the ligand or the receptor, prevent activation of the receptor and ligand binding, and thereby prevent the downstream signaling, which in this case would help alleviate the symptoms of this autoimmune disorder. This is just one example of this all over the body. So in a previous lecture, we saw an example of this with the TNF receptor, for example, with eczema. This happens quite a lot. If we want to turn off some kind of signaling, antibody therapy is a great way to do that if it has a neutralizing capacity like this. Besides blocking, signaling molecules, another thing that monoclonal antibodies are good for is just actually to directly neutralize pathogens as a treatment for infection or even as a prophylactic approach for infection. So an example of extreme relevance right now for this are monoclonal antibodies being produced or being researched as therapies for the SARS-CoV-2 pandemic. So SARS-CoV-2, like most or probably all coronaviruses, they have a pretty common mechanism by which they attach to our cells. So they have proteins on their surface called spike proteins, and these spike proteins bind to a receptor that we express called ACE2. The interaction of the spike protein, the viral spike protein, with ACE2 facilitates their attachment to the cell, which allows them then to enter the cell and establish infection. So what we can do are make monoclonal antibodies that recognize the spike protein, and in particular, the epitope that's really the most important, or an epitope that's really important for the spike protein interacting with the ACE2 receptor. And so you can see an example of that here. If we make neutralizing antibodies which prevent this interaction, now the spike protein no longer binds to ACE2. You basically prevent infection from being established. And this is a great way, again, either usually as a way to prevent the spread of infection, but this could also be an option for as a prophylactic strategy for preventing infection as well in certain contexts. So, this is the case for SARS-CoV-2. This happens with lots of viruses. There are lots of monoclonal antibody therapies for lots of other viruses. But, in this case, we can use it to either neutralize, but also, now that this virus has been bound by an antibody, remember that the Fc portion of the antibody can also initiate effector functions, antiviral effector functions from phagocytes and other cells. So this is a great strategy. This is an expensive strategy. Making monoclonal antibodies in large quantities isn't cheap, but it's one of the many therapeutic strategies that are currently being developed with a lot of intensity for the ongoing pandemic. So we'll see how they pan out. Okay, so these are therapeutic applications. Let's talk a little bit about research applications of antibodies. And in particular, one point that I want to make is that sometimes we actually want to make antibodies against antibodies. Antibodies themselves can serve as antigens depending on what we want to accomplish. And so how would we make an antibody against another antibody? Well, we would basically start with some kind of host animal like a rabbit. We would immunize them with the antigen of interest. So, let's say it's the SARS-CoV-2 spike protein. We would immunize the rabbit. It would mount an immune response to that spike protein. It would make its antibodies. And in this context, we would call this a primary antibody. So it's the antibody made by the original host that's been immunized with whatever immunogen, whatever antigen that we've started with. If we want to make antibodies against these antibodies, we would literally just take these antibodies and put them into a different animal host. And it does have to be a different animal because now this animal is going to not recognize these antibodies. Like, goats aren't used to seeing rabbit antibodies in their bloodstreams. And so the goat now, or whatever secondary host we have, is going to recognize these antibodies as foreign. It's going to mount its own immune response to the antibodies. And so it's going to make antibodies to the primary antibody. And we call these antibodies secondary antibodies. So, we can use these secondary antibodies most commonly for research applications, and they facilitate a lot of different things. And so we're going to talk about some of the options. But again, it's important that we use two different animals. If we just put these antibodies into another rabbit, chances are that rabbit wouldn't respond because, a rabbit has rabbit antibodies, right? And there might be genetic differences in the rabbit. It might be immunogenic, but using two different host species makes sure that we get a really robust secondary antibody response to the original antibody. Okay, so what can we use secondary antibodies for? Well, a common way that we use antibodies in the lab are to visualize things. They help us identify individual proteins of interest or other molecules, usually proteins, that we either we want to see or we want to measure in some kind of experimental preparation. So a really common thing that we use antibodies for is literally under the microscope to see things that we want to see. And ways that antibodies help facilitate this are a process that we call immunofluorescent labeling, or just immunofluorescence, IF. So if you take, for example, a cell and there's some kind of molecule on its surface that you want to see, so and we're going to use that as an antigen. We would usually buy, or we could make our own antibody that recognizes that antigen. So some kind of antibody whose FC domains recognize the protein that we want to see, whose Fab domains recognize the protein we want to see. The Fab is where the variable regions are. But that antibody is going to be bound on its FC domain by some kind of fluorophore, some kind of molecule that glows some kind of color or makes some kind of color that we can see. And so what happens then is if we treat the cell with the antibody is going to stick to the antigen that we want to see. And it's going to then be sort of linked to a fluorescent molecule that we can actually visualize under the microscope. And so this works fine, except one limitation of this is that typically this means that we only get one fluorophore per antigen if we use a directly conjugated fluorophore to the primary antibody. Sometimes this is enough, but particularly if the expression of this antigen is low, if there's not a lot of it, this might not be enough for us to actually see it. We need quite a bit of signal, of light intensity to be able to see it under most microscopes. So a way to overcome this is to use a technique that we call indirect immunofluorescence. So this was direct. So what does indirect mean? Here, our primary antibody recognizes the antigen that we want to see, but we actually make a secondary antibody against that primary antibody. And the secondary antibody is what's coded or conjugated to the fluorophore, to the fluorescent, indicator molecule. Usually we do this in such a way that the primary antibody can actually be bound by multiple copies of the secondary antibody. And so here you can see, as a matter of just ratios, here a single antigen is going to have 3 fluorophores, as opposed to with direct we only had 1 fluorophore per antigen. So here the signal is going to be a lot brighter, and we're going to be able to visualize proteins even that are expressed at much lower concentrations compared to the direct method. So secondary antibodies make this possible, and so this is extremely common in many biological research laboratories, including my own. And so this is an example of one of thousands of pictures like this that I've taken in my career. But here I'm using indirect immunofluorescence to visualize a inflammatory transcription factor called NF-kappa B, which we talked about in a previous lecture. So in this case, I have neurons growing in a dish. And I want to see NF-kappa B expression in these neurons. You can kind of tell they look like neurons, hopefully. So what I did was I bought an antibody that had been raised in rabbit against mouse NF-kappa B. So that somebody, at the manufacturer, literally took the mouse NF-kappa B, purified mouse NF-kappa B. They immunized a rabbit. They took its serum and they purified out the antibody, basically. So I had a rabbit anti-mouse NF-kappa B. So I applied that to the neurons. That stuck to all the NF-kappa B within the neuron, but I can't see that. There's nothing fluorescent there on the primary antibody. In order to visualize that, I then took a secondary antibody, which had been raised in goat, against rabbit IgG. So basically they took the constant region of rabbit IgG antibodies. So this is an antibody that's specifically going to recognize basically all rabbit IgG antibodies, and it was raised in goat. And so this antibody I applied second after I had already applied the primary antibody. So the secondary antibody is conjugated to a green fluorophore. So it's going to fluoresce green if I look at it under the microscope. So any antibody that had bound to NF-kappa B, any primary antibody that had bound to NF-kappa B, is now going to be bound by the secondary antibody, which recognizes rabbit antibodies, and it's going to have a green fluorescent signal associated with it. So when I look under the microscope now, anywhere that NF-kappa B is located within these cells is going to be green. And you can see there's a lot of NF-kappa B in neurons. Like I said, this is extremely common. You see this in the literature, and this is really, this underlies most of the, or many of the advances that we've made in cellular and molecular biology over the past couple of decades. So a very central technique. But looking under the microscope is not the only thing that secondary antibodies allows us to do. We can measure protein concentrations or other antigens of interest using a number of other biochemical strategies. Another very common one is Western blot. Maybe you've done a Western blot either in a different course, or if you're doing research, you've probably at least seen a Western blot in a paper. But nevertheless, if you haven't seen it before, the very simplified version of the way a Western blot works is that we take some kind of lysate, either cells in culture or some kind of tissue that we've taken from an animal, and we take all the protein in that lysate, and we run it out by gel electrophoresis in order to separate it by size. So remember that if we run it out based on its charge, smaller proteins migrate faster, so they're going to be closer to the bottom of the gel. Larger proteins are going to lag behind. They're going to be closer to the top of the gel. So once we've separated them by size, we might want to see a protein of interest. And in that case, we're going to transfer the gel onto a membrane, and the membrane we're going to put antibodies on, the same way that we did in the preparation before. We're going to have a primary antibody that actually recognizes the protein that we want to study. The protein is the antigen in this case. And so the primary antibody is going to bind our protein. Now in this case, the secondary antibody, it might be fluorescent. Recently, Western blots have started to use fluorescent conjugated secondaries, but the more old-fashioned and more common way that this works is that the secondary antibody is actually, instead of being conjugated to a fluorophore, is conjugated to some kind of enzyme. And that enzyme, when you add its substrate, it catalyzes some kind of reaction which causes a color change. In this case, it causes some kind of brown or black color to accumulate, basically. And so wherever that protein is on our gel, in this case it looks like it's about a 50 kilodalton protein, if it's there, the secondary antibody is going to recognize the primary antibody, and when we add the substrate, it's going to catalyze the color change reaction. So basically, the more of the protein that's in this well or in this lane, the bigger the blob you're going to get, basically, the bigger the band, we call it. And so you can see that in this preparation, the HeLa cells have a bit more of this protein than the 3T3 cells in this lane, just because there's more of a color change happening. Again, this is made possible by secondary antibodies, which are linked to some kind of indicator. In this case, it's a chromogenic enzyme as opposed to a fluorescent indicator like we saw before. One additional technique I want to talk about is very similar to Western blot, but usually, so for Western blot, often we're looking for proteins that are either in the cell, like in the cytoplasm, or like in the tissue somewhere. If we want to look for a protein that's actually secreted, and so we would find it maybe in the blood or in the tissue culture media, more frequently there we use a technique called ELISA, or enzyme-linked immunosorbent assay. So ELISA allows measurement of either antigen or actually antibodies in some kind of liquid matrix, the blood, media, and so on. So what if we want to measure the concentration of antibodies? So this is very common if we want to see if somebody has made antibodies to some kind of infection. And so again, with SARS-CoV-2, if you think you might have been exposed and you want to see if you have immunity to SARS-CoV-2, one thing you might do is submit your blood sample to see if you have antibodies against the spike protein. And so we'll use that as an example. If we wanted to measure SARS-CoV-2 antibodies in your blood, what will we do? Well, we would take some kind of a well, take like a tissue culture well, and we would coat it with the spike protein, in this case the antigen that the antibody is supposed to recognize. Whatever that antigen is, in our case the spike protein we'll use as an example. So we're going to attach that to the bottom of the well so it sticks. Then you're going to add your sample, in this case your blood sample, and any antibodies that you have that recognize that antigen are going to bind and they're going to stick. And so the more antibodies you have, the more are going to stick to the antigen coated on the surface. So then when you wash the blood sample out, everything else leaves, but those antibodies stay behind because they're stuck to the antigen that we're coating the well. So what we do then is we add now a secondary antibody that's going to recognize the primary antibody, in this case your spike protein antibodies. And the secondary antibody, again, is conjugated to an enzyme which catalyzes some kind of color change reaction. When we add the substrate then, it's going to cause the well to change colors. Often it's yellow. And we can measure the strength of that yellow color as an indirect measure of how many antibodies we had in the sample. And so, the more antibodies you had, the more are going to be there. The more secondary antibody is going to bind. That means there's more enzymes. The color change is going to be stronger as a result. So a really strong color change means there was a lot of antibody in the original sample. If there's not much color change, that means there was probably none or very little. And so this is a very helpful way for measuring the concentration of any kind of antibody in a sample. What though if we want to measure the concentration of a protein? In contrast, here we would coat the antibody with, or we would coat the well with the antibody, so the exact opposite. And we call this method a sandwich ELISA. So here we would, whatever protein we want to measure, we would take a well that was coated with antibodies against it. And so then we add our sample. If the sample contains the protein that we want to study, it's going to bind to those antibodies and stick and stay behind. We can wash that out now. So now all that's left is our antigen that we're trying to measure. Now we're going to add a secondary antibody that recognizes the antigen on a different place. So this is a case where we want to take advantage of the fact that antibodies can recognize multiple epitopes. So this is why it's called a sandwich ELISA, is because you can see you kind of have an antigen sandwich here. You have the, what we call the capture antibody on the plate. The antigen binds to that, and then the detection antibody binds at a different location. And so the antigen ends up being sandwiched by two different antibodies. Again, the more antigen is there, the more secondary is going to bind. That means there's more of our enzyme present, and it's going to catalyze a color change whose strength is going to be basically directly proportional to how much antigen was in the sample. And so we can use this to measure all sorts of secreted molecules. Commonly in immunology, we use ELISA to measure the concentrations of things like cytokines, either in the tissue culture well or, again, in the blood. But you can use this to measure all sorts of things. Again, ELISA, though, is usually measuring proteins or antibodies that are in a liquid, that are, have been secreted from the cell, basically. Okay, let's summarize. Antibodies can be produced for research and clinical applications, as we've seen. So polyclonal antibodies, which is what we get from a natural immune response, they recognize antigens on multiple epitopes. So there could be many different epitopes that an individual antigen can generate an antibody against. However, monoclonal antibodies recognize a single epitope on an antigen. Making monoclonal antibodies requires the use of things called hybridomas. And so to make a hybridoma, we first must isolate single plasma cells and we merge them with myelomas, those cancer cells, in order to make some kind of immortalized plasma cell line. So now this cell is going to come from a single plasma cell, but it's going to be immortalized, so it's going to divide rapidly and forever and just pump out lots and lots of antibody that all, again, is all going to be against a single epitope on an antigen. It's going to be monoclonal. Antibodies have major therapeutic applications. We discussed a couple of them. We can use them, for example, to block receptor binding for cytokines, toxins, and their receptors. There are a lot of different things that we can neutralize with antibodies that we've made. We can also use them to directly neutralize pathogens. And so, as I said in an earlier lecture in this module, in the clinic, this would be referred to as gamma globulin therapy, just kind of an old-fashioned name for antibodies. But in any case, there's something that we want to sort of soak up or neutralize with antibodies. Finally, antibodies have major research applications. So antibodies can be raised against other antibodies, and we call those secondary antibodies, and they allow us to usually to do some sort of indirect detection of some antigen that we're interested in, usually because they are linked to an indicator molecule like a fluorophore or some sort of chromogenic enzyme. Okay, so that's our first half of the discussion of antibody applications. And the next lecture, we'll talk a little bit more about things that we can do with antibodies in the research lab. See you then.